Okay, well, good evening, everybody. Thank you for coming.
Yes.
And thank you for the people attending online. And it's my pleasure to be introducing the second seminar of the fall from the School of Marine and Atmospheric Sciences here on the Stony Brook Southampton campus. And our speaker is Luis Medina.
Yes.
And Luis is a freshly minted PhD from SoMAS. He just got his PhD defended this summer.
Yeah.
I remember.
In August, yeah.
Yeah, after, we'll just say many years at Stony Brook. I remember having Luis as a student in my class.
Yes.
And Luis comes to us from Venezuela. He did both his undergraduate degree in marine biology there and a master's in biological oceanography.
Yeah.
In Venezuela. And I think did probably some cruise work there with people from SoMAS.
Yeah, that's where I met
Yeah.
Dr. Taylor, Dr.
Yeah.
Scranton.
Yeah. And the topic tonight, microplastics, it's certainly a hot topic in oceanography and marine science. But the interesting thing is that, I don't know if Luis will say it, but from my reading of the field, I'm just gonna be honest, there's a lot of bad science out there. It's because it's an emerging field, and I think there's not a standardization of methods. And so some people are jumping to conclusions that may not necessarily be correct, but what I can tell you is Luis's work is cutting edge and incredibly novel. No one else, to my knowledge, or few other people are taking the approach he's taking. And it's very definitive as opposed to some other approaches, which in my mind are, well,
Yeah.
maybe Luis will explain it for us. So without further ado, I bring you Dr. Luis Medina.
Thank you. Thank you very much, Dr. Gobler, for the introduction and for inviting me today to present my work to you. And thank you for being here with the weather and everything. It's been complicated. But yeah, I've been trying to answer this question: do microplastic contaminant distort understanding of the ocean carbon cycle for the last 3 or 4 years? And I don't know if I answered, but I'm working on that. And we have very good results to show that we are in a good way. And I'm going to, like Dr. Goldberg said, I'm going to show you some approach that is usually not found in the main media or wherever you read about microplastic. This is another point of view of microplastic contamination in the ocean. So I'm going to just keep that. Dr. Goldberg just introduced me. And I'm going to introduce the concept of polymers. Polymers are big molecules, macromolecules that we call. And these big molecules are made from building blocks that we call monomers. So repeat unit of these small molecules create these big macromolecules. And I'm sure you're familiar with some of these macromolecules: carbohydrate, protein, lipids. Those are macromolecules. Those are polymers, but these are natural polymers. You can find those in your body, in the nature. The difference between those monopolymers and plastic is plastics are human-made, man-made. So we create these polymers, which are, for example, polyestyrene, polypropylene. In this case, styrene is the monomer of the polymer polyestyrene. So we create these polymers. And what are the advantages of these plastics, these synthetic polymers? They are lightweight with high strength, so they are very light and they are very strong. So we can use plastic everywhere or in any of our activities. They can be molded in any shape. They are water resistant, they are shock resistant, and the most important one, they are inexpensive. They are cheap to make, so we can make a lot for very few money. So I'm sure you're familiar with this type of plastic, this type of polymer. You can see here how plastic can be found in any or activity, the food industry, entertainment, health, everywhere. We use plastic in every aspect of our life. So since I want to show you this figure, since 1915 to 2015, around 8,300 millions of metric tons have been produced of plastic, have been produced. This is 8,300 millions of metric tons. Some of these is still in use, 2,500. Some of these in use go to the recycle process, but a lot of that is discarded. around 4,900 millions of metric tons are discarded. So here also you can see a projection to the future from right now we are here. So the red line is all the waste discarded. The primary waste generated is this black line. So this black line just telling us that we're going to continue to produce plastic. If that plastic is going to be waste. So this black line, all the waste discarded is this red line. And the important thing here is you can see the trend, how it's going up the part. The green line is, the green line is the incinerated one, the one that we're going to put in the incinerators, and the recycled one. So you can see there is a huge gap between the projection. This is forecasting of plastic in the future. So another figure that I want to show you is this 4,900 millions of metric tons is equivalent at the production of oil, the entire planet production of oil from 1 year. So we have been producing since 1950, the equivalent in volume of oil. So, and if you compare that with the monument in Washington, the obelisk, you can see the difference here. You can see the obelisk here and all the plastic production in terms of volume that we have been generated. So a lot of this volume of plastic, or all this plastic here, is going to the environment as discard plastic. So the big question here is where all this plastic go when enter the environment. So mainly when this plastic enter the environment, reach the coastal areas, because mainly of the river transport. Once they enter the coastal area, they can travel parallel to the coast, they can go to the open ocean, they can sink to the sediment, they can come back to the beaches, or they can be transported by the air and travel hundreds of kilometers or miles in the environment. So they get to the coastal area, and from there, most likely it's going to reach the open ocean. So once they enter the open ocean, they are carried by this current, this huge current in the Pacific Ocean, for example, in the Atlantic Ocean. So from the coastal area, this huge current transport plastic, and they are getting accumulated in the gyres. For example, in the Pacific Ocean, you can see here the Pacific Ocean. Is the west coast of the United States, Japan around here. So we have the California current here. And we're now finding these huge areas of accumulation. And mainly, maybe you are familiar with this in the news, they call garbage island, but they are not islands in the sense that we know an island, Long Island. They are just accumulation areas. So these huge areas where you can find more plastic than in other parts of the ocean. So we have now these huge accumulation areas. But there is another way that the plastic can travel in the ocean, not only horizontal by the currents, but also they can move vertically from the surface to the bottom. And these processes are due by biological organisms that can move pieces of plastic down. They ingest plastic in the surface and then move it down. This process also can be physically. They move the wave, produces internal wave, and then this plastic can reach the bottom. So this is a picture taken by the Japan agency in the coast of Japan. So you can see here the bottom of the ocean here in the coast of Japan, full of debris, plastic and other debris. So once plastic is in the ocean and they reach the coastal areas, they move to the ocean, they are subject to two types of process, biological process and physical process. These tend to break down bigger pieces of plastic into small pieces of plastic in the process that we call fragmentation. From a big piece, we can have thousands or millions of small pieces. So for example, bacteria, biodegradation can do these processes, or also wave, current, and photo oxidation is very common. So when you have a piece of plastic, you live here in Southampton in the beach, and you know that you leave a piece of plastic there on the sun all the summer, the plastic becomes changing color. Also, the texture of the plastic starts to change. And if you touch that surface of the plastic, you can get in your fingers small pieces of plastic. That is fragmentation produced by the sunlight. So the light, the energy from the sun also affect the physical and chemical properties of these polymers. So from these big pieces of plastic, we can get, as I mentioned before, thousands and millions of these particles. So that's when microplastic is important, because microplastics are pieces of plastic smaller than 5 millimeters in diameter. So you can see here a rule, and this everything that fall here and is smaller than this is considered a microplastic. So microplastic can be primary. Those primary microplastics are produced by the industry. For example, in the healthcare industry produce a lot of this beads, and also secondary microplastic that come from the fragmentation process that I just mentioned. Bigger pieces of plastic in the environment produce thousands or millions of small particles. So you can see how diverse here is are those polymers. They are diverse in terms of the shapes. They are diverse in terms of the color and the chemical composition. So it's very, very, very challenging to study microplastic because of all this diversity that you can see here. We're not talking about a unique type of particles. We're talking about different type of chemical composition, shape, size. So microplastics have been detected everywhere, in the blood, salt, water, air, food, even the stool. The research that argue they can be cancerogen. So all this is new research that have been out there recently. So we still don't know how microplastic can affect human health, but that is why we are here to try to answer those questions. But I am very interested in the microplastics in the ocean and what are the effects of microplastic in the ocean to the organisms that live in the ocean, and also to the chemical and physical processes that occurs in the ocean. So I'm sure you're familiar with all these pictures here. You are familiar with this type of image where you open a fish and you find a lot of pieces of plastics here, animals stranded on the beach surrounded by plastic. And this is very interesting. This is a crustacean, very small animal. And you can see here the green fluorescence particles. Those are small pieces of plastic that this small animal have been ingested. So this is not definitely, because these are experiments that have been doing that they conduct in the lab. So these animals are not subject to the same conditions than in the environment. So for this animal to eat all that, they put a lot of those pieces in the environment, in their tanks. So there is a lot of things that we need to understand about this, but they can ingest those microplastics, and they can also affect the food web in the ocean. They can affect organisms at cellular level, individual, and ecosystem. When we go to the beach and we find all those pieces of plastic, it's like, you don't want to be there. You prefer move to another beach with cleaner and that kind of stuff. Also, ecosystem for these marine organisms. So we are trying to still understand what is going on with the microplastics in the ocean. So this is an example of studies that have been doing here in the United States. The national park, the NOAA, together with some universities, they sampled 37 beaches around the US coast, Hawaii, and what they do is surveys of sediment. So they go to the beach, they collect sediment, and they found plastic in all 37 beaches around the US. The most affected beach were the Great Lakes and Pacific Island. And they found around 21 to 221 pieces of plastic per kilogram of sediment. And this is just to show you 1 kilogram is about 2 pounds. It's a bag of flour, 2 pounds. So in that amount of sediment, it varied from 21 to 221 pieces of plastic. So one of the problem is from model estimate indicate that around this number, 5.2 to 5 trillion macro and microplastic particles have been reaching the ocean. So this is the model. This is what modeling people are telling us. 5.2 trillion macro and microplastic particles are in the ocean right now. But the problem is that this only accounts for 1% of the mismanaged plastic that we believe enter the ocean. So in all surveys, all research that we're doing, we only have been found 1% of the plastic that we believe enter the ocean. So in this figure here, the black line is that all plastic produced is similar to the figure that I showed you before. The black line is all plastic produced. And this green dot line is the sulfate plastic, the plastic that we found in the soil. So what is going on? What happened with all this plastic? That is a question that we want to answer. What is going on with all this plastic? The other thing, one of the things that we believe have been happening, and this has been changing a lot through the last 2 or 3 years. We have been found new methodologies to try to address this problem. But at the beginning, when people start to study microplastic, they were using this type of nets. We call this zooplankton net. So these are circular net, and the mesh of this net is approximately 0.3 millimeters. So everything that is smaller than 0.3 millimeters go through this net. So we cannot collect anything as small than 0.3 millimeters in diameter. So all those pieces of plastic, they are very, very small. They are not accountable by this type of study. Okay, so they just collect pieces of particles bigger than this mesh. So we're missing a lot of the particles. We're missing a lot of that mass of plastic. So we think that this is one of the problems. As I mentioned before, in the last 2 years, more people have been doing applying another approach to sample microplastic. But a lot of them, those numbers that you see in the press right now, is based on this type of a study. So we think that an important fraction of the particles have likely been under study. So we don't know what's going on with those small particles. And we're going to see a little bit later why they are important, because you can think, well, but these are a small thing that it's not going to affect me. I don't want to see that, but we're going to see later why those small particles are important. The other thing that we're been missing is all these studies basically address the number of particles. So we go out, we use the net, or we go out to the sediment and we count particles, 200 particles. But the problem is the industry don't, the numbers of the industry that produce the plastic is in terms of weight, the mass of plastic that is producing. So we need somehow to report our funding in mass, weigh those particles, weigh those small pieces of particles. Because we don't know, for example, the process of fragmentation, how many particles produced from one big particle. That is uncertainty there. So the better way to try to understand the problem of plastic is if we can measure their mass. And if you think about it, you go to the beach, you collect one piece of plastic, you can put it in the balance. That is perfect. You have the weight of that piece of plastic. But what's happened when you have these type of particles that you cannot see even you. So you need a method to address and to measure the mass of all those 5.2 trillion particles. So for example, I'm going to show you this. This is a filter, like the one they use for the coffee. But this is a special filter that we use to study plastic, but it's the same concept. So everything that is retained here, we can understand, we can study them. So we collect in this filter around 68 particles of plastic. 68, we count 68 particles. The problem was that these 3 particles that you can see here in the red circles account for 80% of the mass of those 68 particles. So where you can be in a beach and you collect those 68 particles, and you can, in another part of the beach, you collect 3 or 4, but you need to report the mass of this, the mass of plastic, in order to understand the distribution of these polymers in the environment. Not only the number of particles, but the mass is important. So To address all these problems, the sampling problem, the analysis of the samples that when we get, I work on this simple method, or in this methodology. And what I was trying to do is just applying what we know about ocean science, how we study things in the ocean, what we know about that, applying to the plastics. And this is important because using those methods that are already been standardized, so people know how they work, people know their uncertainties, you can address the problem better. Because one of the things that we need to do is producing result. That is true, producing data, but that data needs to be accurate. And if you reduce the uncertainty of that data, it's going to be better. So if you use a standard method to apply applying to the plastic study, you can reduce that uncertainty. So we developed this methodology in which you just collect the samples in the ocean together with blanks. This is important. Blanks are samples. If you collect a water sample from the ocean, you need something that tells you that what you collect is not contaminated, but your method is not contaminated by you. So we collect something that we call blanks. So that blank is collected at the same way that you collect your sample, and that is how you measure the contamination, how you control contamination in your So it's important for microplastic studies, it's important always to have blanks in the field, in the laboratory. That's going to tell you you're doing a good job, you are not contaminating your samples. In addition, all that we wear is plastic, everything here is plastic, everything there is plastic. And imagine that you go to the lab, which are full of plastic. So you need to be very, very careful at the moment to analyze taking the samples. You need to take a lot of blank and analyze those blanks in order to try to control contamination. Then we filter those samples and we do the process that we call chemic oxidation. It's just to try to get rid of all those debris that you can find in the water, especially in coastal water. When the water is murky, like the water is brown or very green, we need to try to get rid of those organic materials, and we use this chemic oxidation step, and that digests all that debris. And then we apply a technique that is based on Raman microspectroscopy analysis, and this is just a technique that is going to tell you the chemical composition of the plastic. As I mentioned before, plastic can be any particles with different chemical composition. So what I mean chemical composition, those monomers that form the polymers can be different. You can have styrene, you can have ethylene, you can have a lot of different monomers. So the idea is try to recognize or try to chemically identify those monomers, because that can tell you sources, that can lead you to sources where these particles came from, pathway, how they reach the ocean. So if you can identify those blue particles that you can find in the beach, those green pieces of plastic, you can maybe reach to sources of those contaminants. So then we can measure the mass, and we can count number of particles, because it's important. We count number of particles, and from there, we measure the mass of those particles. So we can have accurate measurement of the mass of those those particles. Then with that mass, we can compare with, for example, what is the industry of plastic has been produced. So we can do comparison between those, and that is important. So basically, our methodology, I mentioned the problem with the nets, when you go to and pull the nets, tow the net. So what we're doing is using this type of bottles. This is a Niskin bottle. So with the Niskin bottle, you can take samples at any depth that you want. For example, you send the Niskin bottle to 1,500 meters depth, that is very deep in the ocean, and you are sure that you are taking samples from this depth. And that is going to assure that you are taking the small fraction of those particles, because anything that is there that bottle can collect is going to be there, no matter the size. Can be 1 micron, can be 5 millimeters, can be small or big, but it's going to be there. So we use this approach that is a standard approach in oceanography. That is the standard approach when anyone go to study the ocean, they use this type of bottle. So with that, we assure that we're collecting the small pieces of plastics. Then, as I mentioned before, we filtered this. We transferred the water that we collect in the Niski bottle, and we pass through this metal sieve, which is around 300 microns, 0.3 millimeters. Anything is collected here, you can see it. You can take it with a forceps. So then you can take that and identify, a chemical identifier. Anything that passes through this sieve is collected in another bottle and then filtered in these special filters, anodic filter. Then we use this piece of equipment, the Raman microspectroscopy, and this just basically, this is located at SoMAS at Stony Brook, and this, what it tells you is a source of light hit the material, and this piece of equipment is used in material science to identify different materials. So what it does is just a laser, like this laser that I'm showing you here. A laser hit the material, and this laser is going to excite the molecules in that material. That material could be this plastic, that material could be wood, that any material. Those molecules that form the material are going to get excited, are going to get energized. And depending on the vibration of these molecules, if you can think about this is an atom, this is an atom, this is an atom, this is a molecule, could be the molecule of water, H2O. So the molecule of water is going to start vibrate. Independent of this vibration, this specific vibration, are going to produce this type of a spectrum. So the equipment is reporting this type of vibration in this spectrum. So studying this peak, I can understand what type of material it is. It's a fingerprint. So I am sure 100% that, for example, if this molecule vibrates in this mode and produce this spectrum, I'm pretty sure, I'm 100% sure that this is polyethylene, studying these peaks. So what this equipment give me is the fingerprints of the material that been under study. So now I can chemically identify that material. I can know if it's polyethylene, polypropylene, PVC, or any other type of polymer. Then I can count the number of particles in those filters that I show you, and I'm gonna know how many particles I collect in that volume of water. So basically I go into the ocean, I collect water, I filter that water through those special filters, and then I can count the number of particles on those filters. And based on those number of particles, I can extrapolate to a specific volume, let's say 100 particles per liter of ocean water. So then I have the number of particles here, and having the number of particles, this is important, I can determine the size of the particle. This red color here is this type of particles, and it's giving me the spectrum of quartz, for example. This is the green one is polyethylene. So I know the size of this part, I know the size of the particle. Knowing the diameter of this particle, I can calculate the area, knowing the area, I can calculate the diameter. Knowing the diameter of the particle, I can calculate the volume. And because I know the polymer, because the equipment is telling me what type of polymer is there, I know the density of this polymer, then I can calculate the mass of each particle. So I can add all these masses and have not only the total mass of the particles in my filter. I can distinguish between different polymers. I can tell you, or the methodology can tell the mass of the polyethylene, the mass of the polypropylene, the mass of the PVC separately, not only plastic mass. They can tell me the mass of each specific polymer. So now I have a methodology that is accurate to measure number of particles and mass of those particles. And remember, we're talking about very small pieces of particles, very small particles, things that you cannot see with your eyes. So the size of the bacteria. Now I'm going to show you, these are the sites that the only place that I went to the coast of Venezuela in the Caribbean. I went there in 2019. And then I took advantage of other cruises, for example, in the Arctic, in the Antarctic. This transect here from, the this part of from Florida to New York is a transect that we collect some samples there, and also in New York Bight. So I'm going to show you very quick just the results from New York Bight and the Southern Ocean, the Antarctic. So I'm going to show you the results of these two sites. So let's start with Long Island, New York Bight. This is Long Island here, and the samples come from the south coast, the Atlantic coast of Long Island. And these are 9 stations located in the Southwest Sewer District. And in this point here is the outfall discharge of this sewer plant. So the tube come from here and discharge all the water that has been treated around here, around in this drain, in this station. So everything come from there to the surface, from this tube, from the plant. And these are part of the study that Stony Brook SoMAS did a couple of years ago. So they helped me to collect water samples from their site. So this night station here in the coast of Long Island, here, part of the Southwest Sewer District. So what I can tell you about that sample, those samples are, these are different polymers, polyester MPs, polypropylene, polyethylene. We have PET, we have composition of different polyethylene and polypropylene, and this is the mass of all these particles that we find. So 83% of these particles were Teflon, which is this PTFE. Teflon is a material that is used in different activities, but the most common one that people know is for cooking, those pans that are made of Teflon. So we found a lot of Teflon in terms of mass. So we found bigger pieces of plastic of Teflon there. But again, we're talking about small particles. In terms of number of particles, we found that 13% was Teflon again, 70% was polyester. Polyester is the one that we use in coolers or packaging. We have 19 polyethylene here, 19%. But what I show you here is the diversity of these polymers, okay? And also what I'm showing here is the most common polymers, polyester and polypropylene, polyethylene. These are found, I found those polymers everywhere. And this is important because these are the polymers that the industry produce most. So these are the most common polymers that you can find in every activity. Packaging, material, the plastic toys, all those 3 polymers, polyester, polypropylene, and polyethylene. In terms of mass and number of particles, what I'm going to show you here, the colors indicate the concentration of those particles. So red colors indicate more concentrated, more particles. Purple, blue colors, less particles, okay? So what I'm showing you here is, let's start with the particles, number of particles. And remember, this is the outfall. This is where all this water that's been treated for the plant come to the ocean. It's this tube that discharges all the water there. So what we found that east to the outfall, no, west to the outfall discharge, we found more particles. Number of particles increased. And we believe that the arrows that you see here are the currents that dominate this area. So we believe that particles that are being discharged by the outfall are being pushing towards the west. So that's why you find more particles here than here. That is just one of the possibilities that we found there. A lot of, we need more sampling, but because this is just one sample, one time. So this is like a picture of what's happening there. So it's important to collect more samples in different periods and different times, okay? So this number of particles, and then we move here to mass. So you can see that the mass is different here. And this is important because we collect bigger particles here in this station. And even though the number of particles here increased, the mass here, the mass was bigger in this station. So you cannot see a clear pattern about the mass. And remember this, around this area, there is a lot of ship activities, those big ship containers that goes through New Jersey and New York harbors goes through here. Also fishing activities, recreational boating, all that kind of thing happened there. And again, this is just one picture, one survey, one time, okay? Another important thing that microplastic, so the numbers here, look, the numbers here in terms of particles, we're talking about the average of particles that we found in all this water was 40,000 particles per liter. So we found 40,000 particles per liter of water in average. The maximum amount was almost 100,000 particles per liter. So you can see here that is a lot of particles. That is telling you that small particles, we believe that small particles are more common, are more concentrated than bigger particles in the ocean. And that is true when we compare our results with other estimates that use net. So now I'm going to show you the Southern Ocean, the Antarctic. And this is a little different. If you see here, this is horizontal. This is how, if you're looking from the air, the surface of the ocean. What I'm going to show you here is depth. So this is if you are in the depth and you're looking to the water column from the surface here to the bottom here. So in the y-axis, you have depth, 0, 50, 100, and 150 meter. And the lines indicate in the axis or in the horizontal axis, you have number of particles, you have the size of each particle, and you have the mass. So the blue line indicates the mass, the black line indicates the size of the particle, and the red line indicates the number of particles. And what I'm telling you, number of particles are more concentrated in the surface and then start to decrease with depth. So these plastic are more concentrated in the surface. When you go deeper in the ocean, they decrease. And then an important thing with the mass is we found a peak around 100 meters in mass. And also that is related to the size of the particle. Bigger particles we found here in 100-meter bigger particles. So bigger particles are going to give you bigger mass. But one important thing that we can see here is that the sigma, this is density of the water, the black line here in the, and this is telling you how the density of that water mass, the density of that column of water. So bigger light water here, bigger, have a higher density here. So it seems like the particles, the plastic particles are following the same pattern that other particles in the ocean. So that is telling me that we can use this methodology to study particles using these standard methodologies. So this is, again, number of particles per liter. We found in surface water in the Antarctic around 20,000 particles per liter, and how decrease with depth. And you can see the colors here are the polymers, the corresponding polymers. We found polyestyrene, polypropylene, polyestyrene, down to 100 meters. So again, high diversity in polymers, and again, polyestyrene, polypropylene, and polyethylene, the most common polymers found in the ocean. So now I'm going to mention this part because that is the question that I want to answer. That is a question that was in the first slide. If this small particle affecting our understanding of the carbon cycle. So we know that already that these small pieces of particles are important because they can be ingested by a small animal and they can go through the wet food in the ocean and ultimately can affect us. We still don't know that. There's still a lot of research going on about that. But my approach was, okay, but what's happening? What is the effect of these particles in the ocean, in the biochemistry of the ocean, in the chemical processes on the ocean? And one important chemical process that's happened is the cycles in the ocean. So cycles in the ocean, any element on Earth goes through cycles. So the cycles are just the movement of this element. For example, in this case, you can see here a very simple representation of the carbon cycle is how this element goes through this different reservoir. When we talk about reservoir, we're talking about the atmosphere, the continent or the land, and the ocean. These are the big three reservoirs that we can find this element. So cycle in the carbon cycle is how carbon go through the atmosphere into the ocean, and into the ocean, how it gets sink into the deep sediment and go back, okay? It's a cycle. And this is important because this cycle is telling us a lot of the climate, for example, climate change. Studying these cycles, we can better understand, for example, climate change and how carbon can affect the climate and the planet. So what my question is, using this particle, is how this particle not affect climate change, how these particles affect our understanding of the carbon cycle. How measuring these particles can affect our numbers, our budgets of carbon. Are we accounting for this particle when we're making calculation of this, of the amount of carbon or not? So one way to prove this was to take one of the important components of the carbon cycle that we use measure and see if this plastic could affect those measurements. So we took POC, particulate organic carbon, and you can think about POC as any organic particles that is there. A piece of animal, a small piece of animal that just is floating there, we collect that and we can measure particular organic matter. So what I was trying to do is, okay, let's see if the standard method that we use to measure POC can measure the plastic in the ocean. And if that is true, how this measure can affect our understanding of this carbon. So again, this is POC, this is organic matter in the ocean, and organic matter is anything that is alive. So we have dissolved organic matter and we have particular organic matter. So our understanding right now is because all those numbers that I gave you before, the plastic in the ocean, I'm pretty sure the plastic is everywhere in the ocean. We can find those small particles in the ocean. So when we measure POC, particular organic matter in the ocean, are we measuring plastic too? And if we're doing that, how that affects our numbers, how affect that our understanding of this, the carbon budget, and our understanding of the climate change. So this is another way to see it. Organic matter, particular organic matter, and the different compartment in this particular organic matter. And why I am choosing the carbon cycle and plastic? It's just because plastics are material that are made 90% of carbon. So plastic is almost pure carbon, and it's like we adding salt to a very salty soup. So when I take in very salty soup, is because we already put in all this fossil fuel carbon into the environment, and now we are adding this salt into a very salty soup, this pure carbon into the ocean. And again, this is going to affect, maybe it's not going to affect in terms of the environment, but it's going to affect our understanding of this. So very quick, what we did is try to test this methodology, which is very common in all over the world. They use this methodology to measure POC, and this is just to put a sample of the organic matter and a very high temperature, and that is going to give you the amount of CO2 that was there through different reaction in this equipment. It gives you the amount of CO2 that was in that sample, and then you can relate that CO2 that the equipment is giving you to the original amount of carbon in that filter that you took. So you can relate those two, and you can have the amount of carbon that you can have in the ocean. And the problem is that we believe that when we capture plastic together with the POC to the organic matter, and we put that sample together in this equipment, the elemental analyzer, everything goes, and the reporting CO2 is incorporated in the plastic and the natural signal. So we cannot distinguish what was plastic from what's supposed to be there. So that's one of the challenges that we are facing. So what we did here is just, okay, let's test that, let's prove it first. If that is happening, well, then we need to see the consequence of that. So we use sediment from Long Island again, this P station, the Long Island study. So we had sediment at SoMAS in Stony Brook. So Dr. Aller there was very kind and provide us with this sediment sample. And we mixed those sediment with plastic. We put sediment and we mix it with plastic in the lab and created this sample. And then we put the sample in the elemental analyzer, and they give us, for example, the percentage of carbon in those samples. It gave us the amount of carbon in those samples, and it gave us the yield percentage in those samples. So the idea here is, okay, I know the amount of plastic that I put in those sediments. I can calculate how much should I get of CO2. So I know what I put here, and then theoretically, I can calculate what I'm supposed to have. If this experiment follow the theory, I should have the same amount. And that's how I can be sure that the equipment is measuring the plastic in my sample. So for that, I have this simple figure. We have here concentration of polystyrene. So if the equipment burn the carbon from the plastic, this should be increasing here. So this is my theory. In theory, what should I get? The equipment burn that plastic, and this is what I observe. And you can see here that it matched very, very well. So that line here with the dot, black dot, is telling me that the equipment is burning the plastic and together with the POC. So I cannot distinguish. If I have plastic in my sample, I cannot distinguish the carbon from the plastic from the carbon from the natural samples. So that is a problem. Or that could be a problem. Because now I need to see if we really are capturing plastic with our carbon measurement. And this is another of the problems that we can have here. We use the age of the carbon to infer a lot of oceanographic processes like transport and new materials, new organic matter versus all organic matter. So that also can bias our understanding of how old is the carbon in the ocean. And that can lead us to mistakes in our conclusion from some of the processes that happen in the ocean. And at the end, that lead us to erroneous conclusion of climate change, possibly. So based on our own measurements, for example, here, the blue line here is the amount percent of fractional abundance of plastic, and the total is the POC. So this is how much in terms of POC, the percentage of plastic in the organic matter here in New York Bight. So plastic can reach around almost 10% of the particular organic matter can be plastic. So that is what it's telling me is not all that particle or organic particle in the ocean that we're measuring is natural. That can incorporate some carbon from plastic, and we need to take that in consideration when we are measuring and where we're calculating, for example, rates or amount of carbon. One of the other part of the carbon cycle that we took was the black carbon. So we measure black carbon using this method, but it's another story. Yeah. But we also did another type of particles that are pure carbon to try to relate this. But okay, so based on that and all this estimation, these boxes, what represent is the volume of dissolved organic carbon. This is detritus, which is all those particles. When animals die, they decompose, they produce particles, so all that detritus. So we have zooplankton here. We have the amount of bacteria here. This is 10 to the 14 grams of carbon. That is all bacteria. 10 to the 14 grams, that is zooplankton, that are those small animals that I showed at the beginning that ingest plastic, those type of animals. Phytoplankton, there are these microscopic algae that live in the ocean. 4 to the 10 to the 4 to the 15. So if our measurements are correct or close, we believe that the amount of plastic in the carbon cycle could reach around 10 to the 13 grams. We don't know if that is our rough estimate, but this is the first approach to this problem, if it's a problem. So we believe that could be approximately 10 to the 13 gram of plastic that is incorporated through the carbon cycle. And you need to think about this, how this, again, can affect our understanding, our budget, our calculation, and then from there, our conclusion, what is happening. So I want to show you here a little bit of the shift in paradigm. So in this type of study. So for example, in the 1990s, there was no mention of microplastic in any carbon study, no mention at all. 2004, still people talking about POC, possible contamination, but no plastic. In this type of studies around here, they mention oil from the ship that could be contaminated by plastic, by the samples. And then in 2016, people start to study microplastic in zooplankton. 2018, 19, and 20, people start to acknowledge that plastic could be in the ocean and that can be part of this carbon cycle. But I believe that just in August, we demonstrate experimentally that microplastic can be an important component of the organic matter in the ocean, and that make an important component in the carbon cycle. So I just want to show you here some of the future or work that we want to pursue. This and is also how microplastics travel from the surface to the bottom. So I show you in the Southern Ocean, those lines that go surface have more concentrated particles than, but that also show us very interesting features in the water column that we want to study. So we believe that microplastic can be incorporated in these aggregates, and these aggregates are very important because they can aggregate a lot of stuff. They can be like gelatin, so they can attach a lot of stuff, and when they're big enough, they sink and they transport carbon to the deep ocean. So we believe that this could be a process, a mechanism for transport or vertically transport of microplastic. Also, we want to study, or we start studying, the postdoc in the lab is studying this biodegradation of plastic, how organisms, how cells, bacteria can interact with plastic biofilms, and from there, biodegradation and mineralization of these plastics. So here, you can see some of the, these are not from our lab. I just took this from other papers or researchers. But what we believe we have with Raman also is a possible way to identify if bacteria are taking the polymer from the plastic and incorporate it to the cell. We believe we can do that with isotopes. We found plastic that are made instead of carbon. I mentioned plastic is made by carbon, but instead with carbon-12, they are made with carbon-13, which is an isotope, and we can follow that isotope through the inside of the bacteria. So we can be sure 100% that the carbon inside the cells are carbon from the plastic. So if we can prove that, we can identify a specific clade of bacteria that can degrade plastic using isotopes and Raman. And finally, well, not finally, but this is we are trying to partnership with the Peconic, the PEP, to starting next year, try to identification, distribution, and source of microplastic in the Peconic Bay. This is a thing that we are working on. It's not definitely yet, but we believe it's possible that the funding are coming. So starting next year, we think that we can do the Peconic Bay. And we have another big project that we already put the proposal with NOAA to do not only Peconic, but the Sound and other bays in Long Island, studying this identification, distribution, source, and dynamics of the plastic in the bay, how the plastic move using physical oceanography. So yeah, plastic is everywhere since our survival. We found plastic here and everywhere, but I just want to mention this, and I know this is, but plastics are not all bad. Plastic help us to reach some of the important things that as humanity we reach. So for example, plastic packing helps to dramatically extend the shelf life of fresh food and beverage, ship more product with less packing material. So that may be reducing both of food and packaging waste, plastic insulation, sealant, and other building products making homes safe. So we can reduce an emission of greenhouse gases. So these can be positive things about plastic, but we know how all this can end because we don't manage our waste very good. So marine litter can affect aquaculture, health, tourism, fishing, harbors, agriculture, shipping, power generation. So these are the big problems that we need to address with plastic pollution in the ocean. So there is a lot of challenge there. There is a lot of risk and money that can be in risk because of plastic contamination. And at the end, I just want to mention this. We need science to produce data, good data, accurate data, and can lead us to conclusions, to good conclusions. But also only not that, we need communication and education. We need to communicate with the people. We need to educate the people, and with the people, with all of you here learning about this. Policies based on science are the important things. That's what we need. Policies based on science. And this is a good example of how research can lead us good policies based on science. And that's it. I hope that you enjoy. And if you have any questions. Then I know it's a lot, but I was trying to.
That was great.
To make easy.
Yeah. I have lots of questions and comments.
Yeah.
I'm gonna just throw out two observations, comments for starters. The first is that I can't believe that how much plastic is in the Southern Ocean. The Southern Ocean is so isolated.
Yeah.
The reason, like, for example, I just taught this in class on Monday. You took my class, so you know, like, there's iron limitation. There's not enough iron in the Southern Ocean because the winds just blow around that continent and just isolate that continent from the rest of the planet. And yet it's 20,000, it's only half the number of particles compared to right here. So that, to me, that was like shocking. Totally shocking. And then the other thing that was really surprising, but maybe not surprising, is when you look at the outflow, the amount of fluorocarbons you found.
Yeah.
Right, and so PFAS right now is such a big thing in drinking water and coastal waters. The state, I think, just in the last week passed new regulations on these perfluorinated compounds. And that's by weight, the biggest thing that you found in the coastal ocean.
Yeah.
So that's pretty remarkable.
Yeah. And that polymer was kind of, because Long Island was the third site that I analyzed, never found that in any other places. Okay. And when I found it, I was, wow, this is weird. Yeah. Because what we found is basically polyethylene, polypropylene, polyester, all those polyurethane, all those things that we expect to be there. Yeah. But when I found this, it was kind of, I had to double check. But yeah, it was very common here in Long Island.
Yeah. So, and then a question. So, I think, well, I'll let you answer it. I think I know the answer, but you know the answer, so you can tell me. You looked at microplastics, and so that's like maybe 10% of organic carbon.
Yeah.
What about those huge plastic islands in the ocean? And so I guess I'm just wondering what might be the relative importance of the larger plastics relative to the microplastics. But would that tip the scales at all or not?
No, I think in terms of what I'm looking for, in terms of, for example, because we need to see this in terms of consequence. If you have a big piece of plastic floating in the ocean, it's not likely that any organism is going to ingest that. Right. But small particles, first, there are more. And for example, they can go through the or sampling, they can go through the food web in the ocean. So we still don't know what are the consequences of these small particles in the ocean. So in terms of big particles, they are more in mass. And if you take a look of those islands, which are not islands, they're just accumulation areas here, are very huge accumulation areas in the ocean that you can find more plastic. And it's just because of the gyres that accumulate there. But I think in importance, I think small particles, and we're trying to show that they are more abundant, and the implication on any and a lot of aspects in the ocean are bigger than big particles.
My question is more looking forward, or not maybe looking specifically, of course, to the iconic day, but also how do you take what you've gathered here and translate it either to policy or to consumers and who, residents?
Yeah.
I don't know the right word there, but like.
Yeah, that is a challenge. Yes, a huge challenge. The first thing to do is the first step as a scientist, I think, is just go there and try to do research, good research, and produce data. That is the first thing. For example, in the Peconic, there is no data about small plastic or research or papers published about plastic, and especially small plastic. So the first step is go there and show they are there, if they are there. If we can sample, if they are there, show, that is the first step. Okay, they are there. Inform people, inform policymakers, they are there. And part of this project is to try to do that. The first step, go out and do the research and produce data that can be used. And from there, we have to, because part of these projects right now is just outreach. Go to the communities and tell them this is happening in your area. These beautiful waters that you live, that you enjoy, this is what is happening right now. So they are aware of what is going on. And from there, I don't know, try to convince policymakers that these are important topics and money need to be invested in here, in this type of research, and money need to be invested and address all these problems, like education or better, I don't know, system to collect these type of particles in the water plants. That could be good policies that can be implemented. But yeah, I think it's a teamwork. It's not only one group of people, stakeholders that are going to do that. I think it's a teamwork, everyone working in their position and make good things, I think. But it's difficult, it's challenging.
You did a great job, though, taking super complicated methodology and
yeah, that is one of the challenges that we face as a scientist, because we need to train ourselves to try to explain people better what we're doing. Because I think we are very good training to explain other scientists what we do, but when we approach people like citizens, sometimes we are not that good. So we need to train ourselves to do that better, to be approachable, to do that kind of stuff. And I think universities are trying to improve that with students and that kind of stuff, to try to do a better, establish a better communication with residents. That is the only way that we can do this. And with the people have the money. I mean, politicians, they need to understand what is going on. So we need to better communicate with them too, and work together, I think.
What about other methods that, I know there's like for me, I feel like standardization of methods is really important.
Yeah.
I think, to me, you've clearly developed, and it's kind of easy to overlook, but I mean, you developed a brand new method here that is very clearly incredibly robust and information rich. And yet there's lots of other people who are, taking other approaches. So I guess
Yeah.
I'm just wondering, like, when you come up with your method, well, when you have your, how comparable is your data to what other people are just counting big particles?
That is one of the big problems that I face with this. Because our instrument allow us to go very, very small in particles, 1 to 20 microns. That's Raman. FTIR, that is the other approach that people use, infrared, which is very similar. The smaller size particle they can measure is around 20 microns.
And you found that everything was smaller.
And a lot, like 90%, 80% was smaller. So to compare with other work is difficult because it's very, very, especially in the ocean. There are people working with Raman in drinking water, but I cannot compare numbers with drinking water,
Right.
or in salt, or in. So, for example, in Europe, the groups in Germany, for example, they work a lot with infrared. That is their instrument. But that is one limitation. They get into 20, maybe 15 microns, but everything smaller than that, I cannot compare.
Yeah.
So very few other works I can compare. But I had to say that a lot of people are taking this approach, a little bit different than this, but like taking some discrete samples or filtering through the pump of the ship. They just take water from a specific day through the pump, and then somehow they reduce the volume and they do Raman or IT. So that is this part of the sampling process and filtration and chemic oxidation has been very, I think, at this moment is standardized. People are following this step, at least people that work with small particles. Because I cannot compare my numbers with numbers of in that other work that use nettles too, because it's like compare ant population with elephant population.
Right.
So we have more ants. Of course, you're going to have more ants than have elephants. So it's kind of the similar concept. So I cannot compare those
But
numbers.
I mean, the point is that the biomass probably and bioavailability level, you've honed in on what's most important here, but yet probably most of the research that has been done and is still going.
Yeah,
It's focused on the things that people can see,
yeah,
which they, you see, you think it's important, but it's, numerically and by weight, it's not important.
yeah.
So, yeah. And it's surprising, I didn't even realize that the FDIR approach only gets down to 20 or
Yeah.
15, because that also is going to miss a lot as well.
Yeah. So it's kind of difficult. Every time that I know that when I infrared, I use this one.
And everybody's so excited about it, and there's all this research going on about it.
Yeah.
And yet we've seen a lot of it's going on.
And another thing is our definition of microplastic, I think, is wrong. And I think people are trying to address that. Because microplastic 5 millimeters down to 0. But if we talk about millimeters, that is not micro.
Yeah.
Millimeters, my 5 millimeters.
Less than a millimeter.
Yeah, exactly. So it's this, and particles behave totally different from small particles to bigger particles, 300 microns. So I think one of the aspect that we need to improve is just, let's define better what we're studying. So it's macroplastic, microplastic, nanoplastic, and we're going to be able to compare better. So that's another thing. But yeah, like you mentioned at the beginning, I think there is a lot of questionable science in this field. And I don't want to say, but questionable science at this field.
Like it's such a new field. You just showed it. 2016 is, so we're really still at like the birth of this field, and a lot of people were picking up. And anyway, I hope your work gets a lot of attention so that people realize that they
Hopefully,
need to standardize their method.
Yeah, the standardization is one of the things that people are very concerned, and And a lot of paper comes like, we need a standard method, we need a
Yeah,
standard method.
yeah,
But it's kind of difficult to get together. Okay, let's do it.
Yeah.
So, and there is not that much, I would say, meetings or getting together
Yeah.
to talk about this. There is,
Of
it's,
course, it's only been since 2016.
it's new, and
We've had
so-
a little bit of a disruption in meetings.
Yeah. but yeah, we need to do a good job, a better job in doing this.
Well,
It's fascinating.
congratulations
you.
on an incredible.
Thank you.